opportunity for a rather surreal hour in the company of an awful lot of common fruit flies in a variety of moods and the ever so slightly obsessed scientists who are using them to unlock the secrets of life. This is a film about life on Earth, its vast range and beauty, and how we came to understand it thanks to a little bug. Like many good stories, it begins with a journey. New York in the 1870s saw thousands of immigrants arriving to reshape the American nation. An inscription at the base of the Statue of Liberty welcomes the tired, poor, and huddled masses to the land of the free. There is, of course, no mention of insects. But this didn't deter the intrepid beast known as Drosophila melanogaster, or common fruit fly. No one knew this little creature would unlock the secrets of life itself. Without the fruit fly, modern genetics wouldn't exist, and our understanding of life would be limited. 2/3 of our genes are the same as this lowly bug's. Thousands of scientists are watching its every move.
The fruit fly is the most thoroughly understood animal on Earth. We have a deep, holistic understanding of what makes it tick, how it develops, and how it behaves. Most of the deep principles in mechanistic biology were first found in the fly. This will get me in trouble with many of my colleagues who work on many other wonderful organisms. But yeah, the fly is king.
The correspondence between human genes and fly genes is so remarkable. It really does make you understand our commonality that we share. And that means a lot of the things that we want to know about humans, things we need to know about humans if we're going to address questions of cancer or birth defects, we can know them not by directly working on humans or not by trying to figure out from clinical settings what's actually gone wrong, but to try to understand the role of this gene in a fly.
Fruit flies are curious creatures and like to see the world, which explains how Drosophila melanogaster, a native of Europe, got to America in the first place, by hitching a lift on cargo ships carrying rum, sugar, and bananas. It was here in America that the fruit fly became a laboratory superstar.
I actually have seen Drosophila on airplanes, like, from London to New York. You'll see one flit away, and immediately you understand that that's how they got around the world originally.
During its voyage across the Atlantic, this tiny vegetarian stowaway munched on sugar and rotting fruit. Cruising up the East River toward Manhattan was fruit fly heaven, lazing on the top deck, tippling on rum, and doing what they do best, breeding.
I hate to break this to you, but fruit flies have mating on the mind. They don't live very long. Their mission is, of course, to produce many offspring. And the male fruit fly is actually quite a romantic little character. He sings a song of courtship to the female, does an elaborate little dance, and then mates.
By the time a fruit fly is a week old, it's heading for a midlife crisis. So these sex addict insects waste no time and copulate constantly. For 40 years, the fly lay low in Manhattan, eating and breeding, little knowing the role its species would play in the future of science. And then in the summer of 1910, something strange started to happen. Residents in the streets surrounding Columbia University became aware of an unusual phenomenon. Things were beginning to disappear. Just one or two at first, then more and more, until no doorstep was safe from the milk bottle thieves. The man responsible was Thomas Hunt Morgan, the father of modern genetics. His students were stealing the bottles to house thousands of fruit flies. Using this tiny insect, Morgan was about to change forever how we think about life on Earth.
Morgan really started out as a classical zoologist. He had a tremendous appreciation for nature and Darwin. I think he really wanted to understand the complex harmony and the complex interactions among different organisms in the environment.
Charles Darwin had come up with a theory of evolution 50 years earlier. Morgan wanted to test it in a lab on an animal.
He figured that there should be a means for capturing one that would be ideally suited for laboratory culture and for subsequent genetic studies. And so he and his associates put out rotting fruit, old food, anything to see what would be trapped there and what might be easy to grow. My guess is if you were to do that same experiment today, you'd try to do genetics on cockroaches.
So why did Morgan choose the fruit fly? Even in the fly world, Drosophila hardly seems to stand out. It lacks the ghoulish charm of the screwworm fly, which lays its eggs in the genitals, mouth and nose of humans. It has none of the infectious stealth of the mosquito. Unlike the medfly, it doesn't get a kick out of destroying citrus crops. But the fruit fly does hold a definite appeal for geneticists.
They're fast and cheap, and that's what has made them a great research tool. They have an entire breeding cycle within 2 weeks, and you can feed them their entire lives, which is about 8 weeks, for a fraction of a penny in costs. And compared to a lot of mammals that are used for research around the world now, this is probably 100-fold cheaper and faster to use in the lab.
For almost a century, Drosophila has remained the animal of choice for genetics researchers. Its appeal is as addictive for scientists today as it was for Morgan.
So I'm trying to keep flies awake to see what happens to them when they can't get sleep. So they're trying to go to sleep, and whenever they stop moving and to try to catch a little bit of shut-eye, I give them a little gentle tap, and they wake up and they start moving around again. The goal of the present experiments are to find a sleep mutant, a fly that's resistant to sleep loss. And wouldn't it be nice if we could figure out ways that we could stay awake and not suffer any adverse consequences?
After opting for the fruit fly, Morgan instigated a series of insect orgies. He plied them with as much banana mush as they could eat and let them get on with a bit of partner swapping. In 10 days, a single pair can produce 200 offspring, so within weeks, thousands of these randy insects were buzzing happily in Morgan's foul-smelling fly room.
It was apparently a fairly chaotic place. There were huge bunches of bananas in the corner, which they would get from the produce store or from the docks probably, and then mash them down into milk bottles. Everything was filthy. Apparently there were cockroaches everywhere, all over everything. And they would always have their fly stocks with their little cotton plugs in the bottles. And they were just crammed together, working feverishly.
It was not a typical lab as you can see now in the beginning of the 21st century. It was messy and dirty and smelly because people were not taking much time to clean it. They were probably eating and sleeping across the fly rooms.
I think it was probably an awful place to be, but except nobody cared because it was so exciting.
Morgan tested all the fly food himself. When he got bored of banana mush, he tucked into the flies, saying eating the bugs would help him understand them better. He said fly larvae tasted like breakfast cereal. The fly was going to help Morgan solve the biggest scientific dilemma of the day: how evolution happens. There were many competing theories, including Darwin's, but no one had been able to come up with concrete proof. But then, unlike Morgan, they didn't have the mighty fruit fly on their side.
One of the first things Morgan really wanted to test was, in setting up an experimental organism like the fruit fly, was how long does evolution take to do interesting things? So one of their first experiments, Morgan and his associates in the original fly lab back in New York, was to take a culture of flies and put them in a closet and keep them in the dark for a number of generations to see, well, how long would it take for them to lose their eyes? There was really no idea as to how long some of these transitions that Darwin talked about in evolution, how long did it take? Did it take weeks, years, or many millennia? And needless to say, the flies never lost their eyes.
Morgan was looking for a mutant fly. If he could find a little shocker with bits missing, eyes in the wrong place, or different colouring, he would be a step closer to understanding how animals evolve over time. But nature is conservative and mutation a rare event. Frustrated, Morgan started to subject his fruit flies to a range of abuse that would put a South American dictator to shame. He injected acids and alkalis into their gonads, span them in a centrifuge, and stuck them in ovens for days at a time.
The only way that you can understand what genes do or even be aware of a gene's existence is by mutating it, by seeing what happens when that gene is gone. And by looking at the different effects of different mutants, you can gradually begin to piece together how the normal process happens.
It's sort of like if you found a a car with an engine and you were trying to understand how the engine worked, you would probably go, oh, let me take out the distributor. What happens to the car if I do this? But it's the same concept. We're going to be taking out one piece at a time and see how does that affect the functioning of the whole. And then by having all of these pieces, we can then put them back together and maybe get some idea of how do these pieces all work together to make a whole fly or a whole organism function.
Today, work on mutant flies is still illuminating every facet of modern life, including alcoholism. Ulrike Heberlein spends her time getting mutant flies drunk in a machine known as a boozomat.
This is a system that we developed to measure the locomotor behavior, the walking behavior of flies when they are intoxicated by alcohol, ethanol. So these are groups of flies that are just being exposed to air, no drugs. And through this tubing, we have ethanol diffusing through it. The ethanol vapor is now coming through the system and it's getting distributed evenly throughout these tubes. And what you can see almost immediately when flies are exposed to ethanol vapor is they start moving faster. And the computer continues to track each of these individuals at all times. Flies behave when they're under the influence in a manner that it's not very different from the way you or I would behave. If you give them relatively low doses of alcohol, they tend to become disinhibited or activated. They run around faster than they normally would. At higher alcohol doses, they become uncoordinated, they stumble, they fall. And what's, I think, very interesting is the fact that the doses of alcohol that induce sort of the disinhibition in a fly and the doses of alcohol that induce sedation are nearly identical to those that will cause the same behaviors in any rodent or in you and me. As alcohol levels rise, the flies become gradually more uncoordinated and eventually they completely pass out.
By isolating mutant flies that either can't hold their liquor, like Tipsy, or the seasoned drinker barfly, Ulrike believes her work shows that alcoholism has a strong genetic component. For 2 years, Morgan's search for mutants proved futile. He couldn't induce a mutant in the lab, and nobody had found one in the wild. And then one day in 1910, a little white-eyed fly simply flew through the window and landed on the window ledge. Fruit flies normally have red eyes. This little white-eyed critter was just what Morgan needed.
The day the white-eyed fly flew into the lab or was noticed in the lab cultures, it must have been a really big day for their life. They get this spontaneous mutant, this thing that hadn't existed before, and their first handle on that class of genes.
When the first mutant was discovered in Morgan's lab, the story goes that he actually took some of the mutant strain home with him to keep it and make sure that nothing happened to it. And this is something which spontaneously seems to happen to fly people. Because without knowing that story, when I was a graduate student and when I found my first mutant, which had taken me several years to get to, I too took some of them home with me to make sure that I had a safe copy.
Morgan bred the white-eyed fly intensively, and soon the fly room became a mutant equivalent of a nuclear reactor. It was the beginning of a century of ever more bizarre mutant manufacture. But at least some lucky mutants get to party. In this experiment, crack cocaine is vaporized and administered to an unsuspecting bunch of flies.
By understanding the genetics and the molecular mechanisms of how these drugs induce these behaviors, we believe we can understand that phenomenon in higher animals eventually and in humans. When I tell people what I do for a living, I usually get a few raised eyebrows. But once you start talking about the problem of addiction and the genetics of addiction, it hits a chord in almost everybody. I hardly know anybody who hasn't directly or indirectly been affected by addiction.
There were no drugs for Morgan's flies, but there was plenty of sex. He bred his mutants like crazy to try and understand how traits like eye colour were inherited.
I don't think you could be a human on this earth and be a member of a human family or a society and not be aware of genes or not be aware of heredity. I mean, you look at your parents and you look at your children, for good or bad, you can see parts of yourself in both of them. It's very clear that there is in some way an inheritance of qualities from one generation to the next.
Early ideas of inheritance generally took the form of something on the order of the mixing of the blood, as it was called. That it was generally assumed that offspring would be some kind of mixture of the characteristics of the two parents. There were notions that there was a miniature version of the organism, the so-called homunculus, that would be carried perhaps in the sperm. There are classic pictures that show a little man sort of curled up inside the head of a sperm. And things of that sort were attempts to describe how it might come about.
Morgan wanted to solve the riddle of life. To do so, he adopted the experimental method of the Czechoslovakian monk Gregor Mendel, who had spent an unnatural amount of time staring at pea plants in the 1860s.
There is probably anecdotal evidence that there were things like genes, from way back. Farmers and horticulturists must have noticed a tendency towards offspring sharing traits, with their parents. But it was never codified. It was never understood as rigorous laws until Mendel did really careful experiments. I mean, he had the insight to actually perform a biological experiment and write things down, to write down the results. I mean, it's amazing after all these years of farming that nobody thought to do that before Mendel did, in the 1860s and 1870s. It's kind of weird.
Mendel selectively bred pea plants and studied how characteristics like plant size, pea shape and colour were passed down the generations. He deduced that these characteristics were encoded by small particles transmitted from one generation of plants to the next. Morgan's experiments on flies turned this theory into fact. Drosophila helped prove that it is genes located in cells on chromosomes which form the machinery of inheritance. It was a monumental breakthrough.
The day-to-day experiments that those guys did established the basis for all subsequent genetics work. In this country, in Europe, in the world, our understanding of what genes are and chromosomes are traces directly back to that work.
I spent too much time here, too much time doing just this, tapping on flies. Unfortunately, because the room is so comfortable, it's hard to stay awake as you tap on these guys.
It's quite warm, isn't it?
It is. The flies like this temperature. And so what the flies like, the flies get. The fly's king. We do whatever we can do to make them happy. Well, except for keep them awake. Because I'm sure they don't appreciate that very much. That's another benefit of the fly is we can do a lot of different experiments to follow up on hunches and guesses that we might have. Rats and mice are expensive and they require a lot of time and effort, and you want to treat them with a great deal of respect, and you just can't do an experiment because you might have a hunch. You thoroughly investigate it, design an experiment, and then you conduct it. With the fly, obviously we do very careful experiments, but at the same time, if you have a really crazy idea, it doesn't cost anything in time or money to try the experiment. And the answer is sometimes very stunning.
In Morgan's lab, the scientific revolution rolled on. In 1911, his brightest student, Alfred Sturtevant, created the first genetic map. This was the beginning of the century's most ambitious project, to map the human genome. By 1914, Morgan and his team had identified 100 genes. By 1926, Hermann Müller created the first ever artificial mutant by using X-rays.
In the lab, the way that Müller started to develop mutants directly was to use things that could alter DNA. And one example of that was X-ray. X-rays are very powerful beams of energy that when they hit the chemical strand of DNA actually break it.
So we do some pretty nasty things to the fruit flies, and I want to remind the viewer that these are just flies. Okay, we're not talking about people or anything like that. But we feed them EMS, a pretty nasty chemical mutagen that causes nucleotide changes right in the DNA. Zap them with X-rays. So we get pretty aggressive. They're just flies, though. Come on.
The era of the super mutant was upon us, and work on these weird fly monsters could be done at breakneck pace. Over the course of a year, Morgan could trace mutations down 30 generations.
Every 2 weeks, they had the results of their experiment instead of waiting 4 months in the case of a mouse. So the accumulation of research tools in Drosophila has been faster and broader due simply to the fact that they can be bred quickly.
Just as the flies mutate and breed, so, in a way, have the scientists working on Drosophila. Today, just about any genetic puzzle can be tackled using the fruit fly. In France, they are studying sexuality by using a gay mutant known as Voilà.
First, we found that the male carrying this mutation displayed a very strong bisexual courtship. So we call it voilà because it's a contraction of the French slang expression à la voile et à la vapeur. Probably we can translate swinging both ways or AC/DC, because this male really is very excited by other males and females. When you put together a lot of voilà males in a mating chambers, You can see them moving, following each other very quickly. And they get excited both by the male being in front of them, and in turn they induce the excitation of the male following them. So it's kind of also both ways, because they lose discrimination of the male that is in front, but they induce courtship in the male following them. The homosexual male is vibrating his wing, is trying to lick the other male genitalia and thrusting his own genitalia toward the other male, trying to copulate with him. I don't think that anybody can find a gay gene. There are probably a lot of genes that when you mutate them will change male orientation and produce in those males either bisexual or even homosexual behavior.
In the 30s, many of Morgan's students left to set up their own labs, and the fruit fly became the acknowledged superstar of biology departments worldwide. In 1933, Morgan was awarded the Nobel Prize for his work on Drosophila. He didn't attend the ceremony in Stockholm. He was too busy scrutinizing flies. Morgan died in 1945, and at this time, his beloved fruit fly was about to go into temporary retirement. Just at the point when Drosophila was walking tall and looking invincible in the lab, it found itself usurped by a quartet of biological simpletons: viruses, bacteria, yeast, and molds.
The sense was that if you really wanted to understand what a gene was, you needed an animal where you could get your hands more easily on the DNA. You needed an animal, you needed an organism where you could, that would breed even more rapidly than the 10-day cycle of flies. You really wanted to ask the hard questions, hard mechanistic questions of how DNA works, how information is encoded in DNA, that you couldn't really use flies.
For over 30 years, the fly skulked around its milk bottles, still being experimented on, but definitely out of the limelight. Then, in the 1970s, Drosophila was reborn as the darling of developmental biology. In the decade of disco, the fruit fly was suddenly hip again. This was the man who created the new buzz around Drosophila. Nobel Prize winner Eric Wieschaus.
A lot of my life has been spent coming in at 8 and not leaving until well after midnight and trying to get experiments to work, or looking at flies, or trying to figure out how things work. And it never gets boring. It's great. As an undergraduate and in college, I had worked in a fly lab. Mostly it was washing bottles, and I think I'd left that period of my education with the sense that I wanted to be a scientist, I wanted to understand development, but it would be really okay if I never saw another fly in my life. For me, the great revelation was that flies had embryos, and you could look at those embryos and they had all the same features, the extraordinary morphological changes in cell shapes and movements, and the simple egg, and it gets more and more complicated and more beautiful with every hour. And that embryo was in an organism where you could do genetics. So you could actually understand things as well as just look at them. And so I kind of quickly forgot that I never wanted to look at another fly again. If the flies would make those wonderful embryos for me, I was willing to spend the rest of my life working with flies. And I have. Flies are really cool. And so they have nice things. They have, like, in a simple sense, they have a head, and they have legs, and then they have an abdomen. But the most striking feature of the flies are, of course, their wings.
Wieschaus borrowed the biomolecular tools developed on bacteria and used them to investigate how an egg, a single cell, turns into a living, breathing animal. He was hunting for the so-called master control genes.
The eyes are actually compound eyes, so they have made of lots of little individual eyes.
All our cells start out the same before turning into hair, teeth, eyes, and so on. Wieschaus wanted to find the genes which ran the show and stopped us developing eyes on our feet or growing hands out of our mouths. Like Morgan before him, to understand the process, he first had to find examples of it going wrong.
The idea of these experiments where you'd establish inbred lines of flies, we didn't know how many genes there were. We wanted to make sure that we'd made enough mutations to hit each gene at least once. And so we set up something like 40,000 little inbred lines of flies. I think back to this time and say, how could we ever have been so insane?
Wieschaus and his partner were looking for a special type of mutant where all the bits are there, but they're in the wrong place, so-called homeotic mutants, like Antennapedia, which had an extra pair of legs growing out of its head. Or Cockeye, the proud owner of an additional set of genitals where its eyes should be.
We had a microscope that had, instead of just the normal eyepieces for one scientist, there was a bridge and a little prism that was built in so that the two of us could be looking at the same microscope slide at the same time. And so we would sit opposite each other and we would look at these slides after slide after slide, looking for a collection of embryos that showed some defect in embryonic development that we'd never seen before, some mutation that would give us a new insight into the way embryos develop.
Wieschaus identified the genes that stop us developing into horror movie monsters and showed that they were turned on and off by a set of master control genes. These genes line up on the chromosome in a pattern which reflects body shape. The gene for the nose is at the front of the chromosome, followed by another for the eyes, head, and so on all the way down the body to the anus. It seems simple, but it was a stunning discovery.
The interesting thing is that these same genes that were identified in flies not only function in flies, but you can find these same genes in all animals, and you can find them in humans or in invertebrates.
So similar are they that you can knock out a fly's control genes and replace them with a human equivalent, and the fly will still develop quite normally. The little fruit fly had a busy diary in the 70s. In California, it kick-started another scientific revolution, which changed forever how we think about behavior. No one had really bothered studying fly behavior before. It was assumed all bugs were mindless little robots. Now Drosophila was going to prove everybody wrong. This experiment separates out flies depending on the strength of their sun-seeking urge. Fittingly, it was developed on the sunny west coast by Seymour Benzer at the California Institute of Technology. Benzer proved that these kind of behaviors had a genetic basis.
Benzer's approach was self-conscious and very revolutionary in that sense, because his idea was that if you isolated mutants affecting specific behaviors and found out what those genes were, then you could eventually begin to understand the brain mechanisms behind the behaviors. And that has proved to be an enormously effective approach.
Behavior was thought to be something almost mystical that occurs only when you've got an entire animal put together and functioning normally, right? That behavior was an emergent property from constructing an animal into its adult form, for instance. And as a result, it was never thought that you could affect one gene and have that effect so severe that you could produce a defect in a behavioral outcome.
The puny little fly sent a huge shockwave crashing through the scientific establishment. If you could mutate flies and alter their behavior, maybe we too were genetically pre-programmed.
The basic idea is take a bunch of flies, put them into a little tube, and put that tube at the beginning of the maze here.
This experiment confirmed the genetic basis of some behaviors. Given the choice of going up or down, fruit flies tend to go up. Benzer bred a strain of flies that did the opposite. Here the two are mixed together. The normal strain go up, the mutants go down. They separate out, leaving the mutants in the bottom tube. Scientists started to think that genes were like light switches and could be turned on and off, altering behavior. We don't have 6 legs or like hanging around dustbins, mostly, but the belief also began to spread that the fly was the perfect model for understanding human behavior. In the 80s, attention shifted to complex behaviors, like memory. Tim Tully has been obsessed with memory ever since he had a sledging accident while playing chicken with his schoolmates.
So I lost one day and wound up going over the cliff head first and really dinged myself bad. Knocked out teeth, bit off my tongue, put a couple holes through my lips. You know, a real mess. And then I became cogent, and my grandmother was standing there looking at me lying on the bed. And I said, "Grandma, what are you doing here?" And she gave me one of these do-do-do-do looks like, "Well, don't you know?" I had no recollection of the previous 2 weeks, none. And it has never come back to me. That knock on the noggin completely erased 2 weeks of my life, a total blank slate.
Tully has devoted his life to researching memory in the fly. He helped to prove that Drosophila is not a brainless pinhead. Using a device known as a mimometer, he demonstrated it actually has an intellectual streak.
One of the first things they do is orient toward odor. So we trap flies in a chamber, and we pass odors through the chamber on air current. And in that chamber, they're walking on a floor that can be electrified, so they get foot shock when we want them to. And what we do then is pass one odor through the chamber and shock them on their feet. So this is punishment in the presence of a particular odor. And then pass a second odor through the chamber without shock. So they learn something different about 2 odors. One's bad, one's not bad.
Using such Pavlovian methods, Tully's research showed that flies could learn to avoid the odour they associated with being zapped. It takes only 3 or 4 sessions for a fly to pass this type of test. Larger, fluffier animals can need up to 30.
At the level of Pavlovian learning, flies actually do it as well as we do. They show all the same basic properties of Pavlovian learning as a human or a dog or any other animal. And in that regard, they appear to be just like us. Now, can they do calculus? No. But that doesn't have to do with the basic mechanism of association. That has to do with how complex your circuitry is. So it's kind of like flies are Philco radios and humans are Macintosh computers.
In 1995, Tully found that by giving flies an extra copy of a gene known as CREB, he could create mutants with photographic memories.
This was the first experimental demonstration that a genetic manipulation could create memory better than normal, photographic memory. So I think the future holds that we will have study aids. If you want to learn to play a musical instrument or a new piece on a piano, pop the pill and you can get those motor skills down in a few sessions instead of weeks and weeks of practice. That's the potential that is suggested by these inklings that we're starting to understand now.
The Neurosciences Institute in San Diego. Once the fly had impressed scientists with its insect IQ, they realized it could be used to chart the intricacies of the human brain. If you can understand elaborate behaviors such as aggression in a fly, you're halfway to comprehending them in us.
What we're looking at here is a close-up filming of flies in a cage. And the way the flies are set up to see aggression is you need two males in a little cage with a territory and one female fly. And one of the males will typically try to control the territory. And the reason is that the female lays eggs there, and that gives the territory value for the male. Notice that the male is coming up. This is the aggressor. He's going to kick the male and continue to kick him until he leaves the territory. And so he stops because the male left. Sometimes the aggression is more intense, and it's not enough that he kicks him off the territory. He follows him into the cage to make sure that he doesn't come back. In some cases, the male that is being kicked off actually resists, and this is a fight that you can see here coming up. The male that is being kicked actually resists. He flips his wings up and will continue to receive kicks, but will not leave. And this is when fights escalate, and you can get some intense boxing and tussling going on, as you can see right now here. So the aggressor actually has his wings up, which is called the wing threat behavior. And now they're fencing, they're boxing, because he doesn't want to give up. He's pushing him, and it seems at some point that the aggressor is actually being pushed away. You can see he's almost shoving him off the territory as a sumo wrestler. But he'll turn back, kicks him again, and now they almost tussle over, and finally he kicks the male back off.
Herman is trying to isolate aggressive flies and see if they're genetically different from the rest.
They're not stereotyped. They don't just do the same thing over and over again. They decide when they want to do a particular behavior and when they're not going to show the behavior. They're quite individual. Every time I see an intense fight, I still smile. It makes me still. I'm fascinated by it. It's wonderful.
Why?
Maybe because I'm weird. I I don't know. Because I think I have a sense that I'm getting closer. When a mutant looks like it might be more aggressive than my background strain, I think I'm getting closer to an answer. And the answers down the road, identifying a mutation in a gene that might play a role, is absolutely exhilarating.
There's usually always one or two very recalcitrant flies in the bunch that really want to sleep, and they're the ones that are hardest to keep awake.
Are you getting bored of tapping yet?
I'm definitely bored of tapping, but I've been bored for several years now. Well, when I discover the function of sleep, then I'll stop. So I'll probably be doing this for many, many, many years to come.
Are you looking forward to that?
Well, you know, it's really an exciting question, and I hate to put myself out of work. So as long as I can keep on addressing this issue and trying to solve this exquisite mystery, I've got a job. Sitting in the dark tapping on flies is not really how I would choose to spend my time, but the results are worth it. And sometimes there's just no other way than the hard way.
The more the fruit fly is studied, the more useful it has become. In the year 2000, it became the first complex animal to have its genome completely sequenced. Scientists have now mapped each of its 13,601 genes. This was done as a warm-up for sequencing the human genome, and threw up a startling result. We have more in common with flies than anyone ever imagined. 2/3 of our genes are the same.
People thought it would be a more drastic difference between both organisms, and in fact they realized that fly was a kind of miniature human.
It looks as though the animal was invented once, a long time ago, and evolution has been very parsimonious in keeping a core set of genes that are involved in very basic cellular processes, whether it's the development of the animal or even the way the brain functions.
We think of flies as smelly, dirty disease carriers. But this humble bug may help find a cure for cancer, Alzheimer's, and Parkinson's. Here in San Francisco, the biotech company Exelixis has joined the Drosophila gold rush. Today, there's big money in fruit flies.
At Exelixis, we have millions of flies, certainly. I don't know the exact number. We have over 25,000 different strains. If flies were as big as mice, we would have to have a building many times this size just to house them all. What we want to achieve with Exelixis is to discover and develop drugs that will provide better therapy for patients. That's the bottom line, that's the goal. So the challenge has been to work one's way from the genes that are increasingly known to be involved in causing the disease to other genes which might be the targets for new drugs that would cause the disease to be treated. And that's been a very difficult connection to make, and that connection can be made actually quite quickly in simpler organisms.
Here they have industrialized work on Drosophila. Every day, thousands of potential drug compounds are synthesized and tested on batches of fruit flies. We've come a long way from Thomas Hunt Morgan and his banana mush in milk bottles.
The important difference between us and an academic institution is to a large extent a matter of scale. We've been able to invest significantly not only in people but in other technologies that enable us to do things in a matter of months, which in many instances in an academic laboratory would probably take a decade or more to complete.
You know, the question of how would Morgan react were he to come here today, I think is a little bit like asking how would the Wright brothers react if they were to go to the airport and see a 777. They'd understand this is an airplane, they understand the basic principles of flight, but the differences between their first primitive plane and the planes we have today are overwhelming. When I look at a tube of flies, I am constantly amazed by the complexity of those organisms. You think about flies as these silly little things that buzz around in a rather mindless fashion. But when you understand the complexity of the genetic systems and of the biochemical systems and of the regulatory systems that are involved in a fly, I think it is truly humbling.
Oh, it's exhilarating to think that you can take and translate such fundamental information, so abstract in its own way, and think about having an impact on disease. I am 100% confident that the work that we're doing today will lead to a new therapy for cancer or for some other major disease.
The work begun in Morgan's shabby fly room goes on. After almost a century of scientific breakthroughs, the fruit fly is still king of the lab. It has made the journey from zero to hero and established itself as the undeniable superstar of genetics.
The future for fly research is driven by the same features that drove Morgan to choose flies at the turn of the century, that you have big questions that you don't understand in biology. Right now, a lot of those questions have to do with human disease, maybe. Turn of the century, they had to do with inheritance. But big questions are hard to answer. And as a scientist, you have to choose a system or an animal that is easy to work with and provides opportunities for you to do the right experiment quickly and easily.
Time just drags. You sit here all alone in the dark room, knowing that in a few minutes, someone's gonna walk through that door and relieve you of the monotony. The last 5 minutes last forever. And you wait for that door to start turning because you can hear it. You can actually hear the feet in the hallway walking past, and you hope that it's a person coming in to save you from tapping on flies. It's always disheartening when the footsteps fade into the distance you know is somebody else. But as they get closer and closer, and you hear the door start to turn, you know your time is at hand.
Could making friends with paedophiles when they come out of prison stop them reoffending? Next tonight, news of an incredibly controversial scheme pioneered by a group of ordinary Canadian citizens, which is having extraordinary results. No more victims in a moment.
Just press red.